Yeah, thank you so much to Houseman's Bookshop for having us, and to Michael for inviting us, and for all of you for being here on a Monday evening. So we're going to talk about the book, but it's been out now for 3 years, so we've also kind of come some kind of way in our thinking since then. But broadly speaking, we're going to be talking about the increasingly important role played by genomic biotechnologies, which is a mouthful, but we're going to get into kind of what that means in a bit in contemporary neoliberal societies. So we're going to try to keep it as informal and kind of interactive as possible. We'll read some bits from the book and kind of contextualize it, but we also want, to have a conversation with you all, because that's the only way that we can kind of think through some of these topics together. So we decided to structure the session in two parts. First one, where Amedeo will be kind of talking us through the kind of broader sweeps of what these technologies are, what they do, what the regulatory framework has looked like for the past few decades, to then briefly summarize what's kind of new, what are the newest technologies that have been kind of causing all of this buzz in the last decade. And after that, so that'll be around 10 minutes, and we'll have a kind of discussion break where also anyone can ask any clarifying questions. These are really dense kind of technical topics, so it's really normal that a lot of the terms that we might use kind of interchangeably might not be immediately clear. So we want to make sure after that intro everyone has a kind of chance to check in if everyone's on the same page. So yeah, in that first part, we look at how we got to today, the history of biology in the context of biological technologies aimed at modifying living systems. And we're going to try to situate that development within a capitalist system geared towards accumulation and exploitation. So, and then we'll have a second part which will focus most on the agricultural chapter of the book. So while the book covers many themes, we thought that was a bit too much to try to go into all of the different fields. So we want to focus on agriculture, also because we have someone here today from GM Freeze and from Seed Sovereignty that can share a little bit about their work in that area. So for that, we'll read some sections from chapter 5, Molecular Factory Farms, and to situate this in a longer trajectory of imperial practices in agriculture, kind of geared towards expanding capitalist agriculture. In particular, we want to focus on the agrochemical industry, and we'll share some of the historical and most recent developments in that field. So before kind of getting into the nitty-gritty, we're kind of curious to know what the baseline of kind of knowledge about these topics are in the room. So maybe if you can just turn to the people next to you and have a quick check-in of do you know what genetic modification means? What are these technologies, and do you already kind of have some idea of how they might kind of connect to broader systems of capitalism? And then just it's just for us to have an idea of where we're at in the room. So if we can take a minute to talk and then let us know, and I'll ask you the question in a bit. Little icebreaker. Sorry, guys.
I don't even know if I know about it. But yes.
Should I read the...
Yeah, I think so. If you feel like it.
Yeah?
Yeah. Maybe I can introduce it and then you can read it if you want. Yeah, do you want to do that? Oh, yeah. I think you can call them into...
Okay, so can we get a quick show of hands? Who does...
That's absolutely fantastic. I wasn't expecting.
Who feels like they know what genetic technologies are more or less already? Kind of. Who thinks this is completely new to them? Who feels completely like, I have no idea what this is?
I know.
Okay, the few. Okay, so we have some kind of basics for what it is, what the technologies are. Obviously, we're going to recap that in a bit. What about, do people kind of feel intuitively that they have an understanding of how these technologies might fit in with capitalism, colonialism? Yeah? Okay. Should we dive straight in then?
Yeah, absolutely, yeah. Yeah, and I think it's good to start from like where the book starts. That we have like this little vignette at the beginning, and I think that was born pretty much from the fact that I think a lot of debates, especially political debates on biotechnologies, starts from the questions like, oh, what if something goes wrong with these technologies? What if something unexpected happened? What are the unexpected kind of events that can go wrong? And obviously that's the real questions. There are all kind of like off-target effects, technically are called, with these technologies. But I think for us, the main question is like, what if these technologies go right? What is like the future that is envisioned by biotechnology companies and kind of tech utopians, and are embodied in these technologies. So like this piece was trying to imagine this future, but like all of the kind of innovations that we talk on are actually then talked about in the book. And obviously are innovations that have not been scaled up, but they exist in some form. So I don't know, maybe you
yeah, so I'll read this kind of pre-part of the book. It's a little bit long, and our editor said it gets very grotesque very quickly. So I'll just leave that there. But we're not trying to only kind of, in having this discussion about what we call the fragments of tomorrow, kind of the utopias for them imagined by the biotech industry, kind of what that would look like, but then what we want to think through together in this room is kind of what other ways can we imagine of kind of dismantling some of the structures we might talk about. So I'll read this bit. It might take a couple of minutes, and then we'll take it from there. In the biotech utopia conjured by venture capitalists, entrepreneurial scientists, and techno-plutocrats, monoculture plantations populated by genetically modified crops cover most of the earth. Row after row of perfectly identical corn stalks producing pesticide in their own cells. Fast-growing sugarcane shooting to full height in half of the time of its unmodified siblings. Herbicide-resistant vegetables prospering in the midst of ever-increasing amounts of poison in the air and soil. Spring is silent, no insects dwell in the fields. A cocktail of bioengineered pesticide-producing plants and genetic extinction technologies have wiped them out. A few lonely genetically modified honeybees, engineered to withstand the pesticides dumped on never-ending fields, buzz among the rows of identical plants. In a nearby factory farm...
The apocalypse.
The apocalypse. In a nearby factory farm, genetically engineered cattle grow bodybuilder-type, hyper-muscled bodies. They no longer have pesky horns that might damage workers or profits. These have been genetically removed. The space is overcrowded, but the veterinary doctors are confident that the last round of genetic modification may contain the next epidemic. Up in the hills where salmon used to swim in the rivers before they were all dammed, sit massive tanks in which the fish grow to maturity in half the natural time. In the rapidly shrinking forests, bioprospectors are busy mining a disappearing biodiversity, hoping to sequence their genomes for the latest proprietary pharmaceutical commodity. Using DNA barcode scanners, they create a digital copy of the forest in server farms located thousands of kilometers away. Timber companies are busy chopping down poplar trees disposed in neat geometrical patterns. These gene-edited trees grow fast, their accelerated life cycle boosting the expected rate of profit. Yet no animal would munch their bark, because the gray squirrels who used to eat into loggers' profits have been genetically sterilized and wiped out. At sea, genetic life support keeps corals breeding in a warming ocean. further beyond the global dead zone spreads. There, floating farms grow genetically modified algae in water that is too oxygen poor to host marine wildlife. Oil rigs in the distance keep pumping, but ships are already releasing gene-edited microbes to lap up the oil spills. Beneath the waves in the deep time of the Hadal Zone, unmanned vehicles hunt extremophilic microbes by hydrothermal vents, sequencing their genomes in search of potentially valuable genetic traits to patent. In crowded cities around the world, conscious urbanites have a choice between a variety of bioengineered foods. This is an actual ad. Become the envy of your friends and followers with this highly sought-after delicacy. Pink glow will look phenomenal on whatever social media platform is in vogue by the time you read this. It's a pineapple. Biofortified foods produced by billionaire philanthropies are offered to the many who have been forcefully removed from the land. In tiny rental studios, miniaturized pigs keep company to the housebound workforce. Capital's internal contradictions appear temporarily suspended. No need for radical social and ecological change. So this section is kind of before the introduction, and we're kind of trying to paint the picture of, as Amedeo said, what is the vision that is being peddled by the biotech industry. So I don't know if you want to maybe start us off by...
Yeah, I guess also you can feel like that the book was written during the pandemic, and I take the opportunity to thank Erika for like making those months much more interesting than they would have been otherwise, like dwelling in and thinking about these topics. And essentially I think I'm gonna like briefly talk about the first part of the book. And the first part of the book, essentially what we are trying to do is to exactly think about the history of this history of mutagenesis, of like genetic modification technologies, or in general like biotechnologies, and how that history somehow was strictly connected with the history of capitalism and the history of this called Anthropocene, or like the ecological crisis. I think I think there were two starting points for us. On the one hand, to start from the idea kind of like that you can think about world ecologies as essentially being continuously formed by the metabolic interactions between organisms. So organisms continuously digest and destroy. No, they are both catabolic and anabolic forces. No? So they destroy and they create. No? And like the biosphere is the result of this continuous metabolic exchanges and metabolic interaction between living beings, bacteria, plants, animals, human beings. No? So you can think of the world in these ecological terms, but also like in the history of capitalism. Now, capitalism, if you take agriculture, I mean, obviously, especially like the critiques of capitalism, the history of Marxism, is very much focused on the factory as a site of production. But actually, if you shift the focus, as Marx himself sometimes does, to agricultural fields, forests, fermentation tanks where wine is fermented that you are making today. And then you see to what extent kind of like the operations of capital are dependent continuously on this metabolic process, this interaction with the other living organisms, being a yeast in fermentation, or trees in forestry, or crops in agriculture, and so on. And therefore, what we explore in the first chapter is the way in which corporations in all of these fields have always been interested in, first of all, understanding these metabolic interactions, understanding biology, and on the other hand, understanding how to control and how to accelerate, how to expand, how to direct in useful way metabolism. So the process of transformation that all living beings must put in play in order to survive and reproduce and grow. And essentially what we look at, I mean, if I have to summarize this history, I think like we look at specifically like mutagenic technologies. Now, mutagenic technologies are technologies that induce a mutation in the gene. So molecular biology usually has like one fundamental kind of understanding of metabolic processes, by which essentially, okay, you have the genetic code, if you want, DNA, that instruct the production of RNA, that instruct the production of proteins. And that's like kind of like the fundamental kind of like understanding of metabolism. So mutagenic techniques essentially are techniques that try to induce or that induce mutations in the genome in order to then transform the way in which the living organism not only physiologically appears, but especially how it metabolically interacts with the environment. So I think it's going to become more clear as I explain it. But I think like the first, like the starting point of all this program of research, it comes roughly like in the 1920s. So we are actually, I was even thinking, one could write a history of mutagenic techniques. It's called the Mutagenic Center, because it's like the first kind of like research in the field is from 1927. So we are almost like 100 years of research in the field. And the first kind of research and one of the first articles that discuss these possibilities published in 1927 by Hermann Müller, that is this biologist, molecular biologist we would say today, but the term wasn't even around at the time, that essentially was dedicated to the study of Drosophila melanogaster, so like the fruit fly essentially. And they discovered essentially that radiation was able to kind of like accelerate the natural rate of mutation of these organisms. So all living organisms have natural mutations. Mutations happen all of the time. Being exposed to radioactive material accelerates that rate of mutation. So more random mutation appear in a shorter period of time. And immediately, already by the end of the 1920s, this was taken as like something. It had two effects essentially. One that was like cultural, if you want, and immediately stimulated kind of like these visions of kind of like essentially what we would call today biotech futurism. And like Müller himself, after winning the Nobel Prize for his research, kind of turned into like kind of a sci-fi writer and writes like Out of the Night. That is kind of like interesting, kind of like, he was also a Marxist and then he moved to the Soviet Union. I mean, he has an interesting biography. But yeah, it stimulates kind of like these ideas that the New York Times kind of publish. Okay, this is like the first step into, is the first tool that will eventually allow us to kind of like control evolution and so on. And then it has more kind of practical implications. Now, in the sense that it starts a whole program of kind of radiation braiding that it still exists today. And essentially by exposing plants especially to radiation, you can essentially create a automated kind of factory of mutant production. Obviously, most the mutations being random most of the time kill the plant or like produce mutations that don't have physiological effects. Those effects are not even clear, but like sometimes it creates like mutations that can be exploited industrially, like kind of a peanuts with a tougher hull that can be kind of mechanically harvested, for example, was created in this way. And there are other examples. so this was kind of like the very origin of this program. But kind of like the biotech industry really takes off only like in the 1970s, with the introduction of recombinant techniques, recombinant DNA. So once again, and this I think is significant now, is yeah.
Maybe should we explain what recombinant DNA is?
Yeah. Do you want to go with this? Should I? Yeah, recombinant DNA. No, I was just saying, once again, is American research. So a lot of the research from the 1920s to the 1970s is concentrated in the US and in Europe in part, but especially in the US. It's significant in part that a lot of funding for this research comes from especially the private sector in the US, and obviously there is a lot of historians that have worked on kind of the influence of the Rockefeller Foundation, for example. That interesting, even the term molecular biology actually was created by the Rockefeller Foundation as one of the fields to which it would dedicate a lot of its funding. And it is essentially from this type of research that emerged recombinant DNA technologies. Essentially, they emerge from the study of bacterial immunity, no? Like the way in which bacteria defend themselves from viral invasion and pandemics, essentially, or epidemics. Also, bacteria can be affected by viruses, and they have like their own immunitary systems. Essentially, what microbiologists kind of discovered around the 1970s is that bacteria utilize these enzymes, no, that are called restrictase enzymes, to kind of like cut the DNA, kind of like destroy the DNA of invading phages or viruses, and the ligase enzyme, that are essentially enzymes that are used by the bacteria in order to repair its own kind of DNA. No? So like these types of enzymes, essentially the idea was to use these enzymes and repurpose them in order to cut and paste DNA. No? So that's why today we talk about like transgenic kind of organism, in the sense that like these early technologies essentially were able to kind of like cut fragments of DNA from one organism, now using restrictase enzymes, and like paste it into kind of like the DNA of another organism. And it did indeed, I mean, an early name for like associated to what we call now genetically engineered organism or transgenic organism was like molecular chimeras. I mean, actually, that there was the technical name that was used by Cohen and Boyer. That were the two people that also won the Nobel Prizes for this research. That immediately was thought to have kind of like practical application. Indeed, Boyer himself, one of the inventors, after depositing the patent on recombinant DNA technologies, he founded a company that was called Genentech. That generally is understood to be like the first proper biotech company. No? And the name itself of Genentech was like a kind of like play on genetic engineering technologies. No? And Genentech would go on essentially utilizing this technique of recombinant DNA in order to essentially work with bacteria. No? And the most successful kind of like product of Genentech already by the end of the 80s was to work with Eli Lilly, that was like a major pharmaceutical company, is a major pharmaceutical company, to produce like synthetic insulin. No? And essentially what they did is like they spliced, that was the technical term, kind of like the gene that encodes kind of the information for the production of human insulin in human cells into Escherichia coli bacteria, and that way they induced Escherichia coli bacteria to produce, to metabolize insulin as part of their life. I think it's interesting in the sense that it kind of captures what genetic engineering is always about. You know, it was about kind of like inducing kind of an organism like Escherichia coli that goes along, has his own life somehow. Now it eats and it kind of like transform this feed in what it needs to survive and to grow and to multiplicate, to have a different life. Now to feed, but to then use this nutrition to kind of do something else and to produce something that wouldn't otherwise produce. But essentially, the big kind of innovation that came from recombinant technology was mostly in agriculture. I mean, there were other recombinant vaccines were another big one in the pharmaceutical, but then agriculture with the introduction of herbicide-resistant crops and pesticide-producing kind of crops in the early 1990s, of which Erica will say more about in a bit. So this kind of like the background in which and kind of like recombinant technologies until essentially 2013 remained kind of like the fundamental technology on which basically all of the biotech industry was based. No? In 2013, essentially, there was like what was generally recognized to be like the next major breakthrough. There was the introduction of like these CRISPR technologies or CRISPR-Cas9. That also won the Nobel Prize in 2020. And that, once again, first of all, created that immense hype in the biotech industry, created a new kind of wave of investment in biotech. Billions of dollars were invested after 2013. And essentially it was because of the introduction of a new way, again, of introducing mutations in organisms, so manipulating the genetic code in order to transform the metabolism of bacteria, plants, and animals used in different industrial sectors, from pharmaceutical production to agriculture, to the livestock industry, to aquaculture, and so on. So what's different between CRISPR and recombinant DNA? I mean, it's difficult to give like a straightforward answer. I mean, I think the book does a much better job than I can do now. But essentially, like CRISPR itself, despite the name, the fundamental element in CRISPR is essentially Cas9. It's like the Cas enzyme. No? So just like recombinant technology were based on bacterial enzymes, now restriction and ligase enzyme, CRISPR essentially results from, once again, research on bacterial immune system, the biology of bacteria, and once again the finding of another way in which bacteria fight off viral infections. That is this Cas enzymes. And Cas enzyme, once again, work pretty much as, in fact, they are a form of restrictase enzyme. They destroy the DNA of viruses, but the difference is that they are guided by a piece of mRNA in where they hit the DNA. So what does it mean for the biotech industry? It means that then Jennifer Doudna, who won the Nobel Prize, essentially wanted to, because she was able, and her research team was able to essentially appropriate this process by giving a piece of mRNA to the Cas enzyme, essentially to direct the Cas enzyme, to tell the Cas enzyme where to cut the DNA. So essentially, they are generally presented in journalistic discourse as kind of like genetic scissors. No? They cut the DNA where you want. Now scientists are able, or biotechnicians are able to kind of synthesize a piece of mRNA to tell the enzyme where to cut the DNA. The final effect of that is, okay, you have a tool that essentially, instead of just allowing you to transfer DNA from one organism to the other, it allows you to modify the DNA pretty much as you want. So it's much more flexible, it's much more universal and it's much cheaper. Now, you can make much more experimentation with it at much cheaper prices. So it creates kind of an acceleration in industry, and a new wave of mutant kind of organisms came out of this innovation. And I mean, to conclude, no, I was thinking. I mean, it's difficult to capture really like kind of the political side of this, and so I thought I would take this passage that we take from an article that was written by Bill Gates, if anyone. Bill Gates in 2018 actually, to kind of like explain his investments, both kind of like financial investments and kind of charitable investments, and also political kind of discursive investment in this technology, publishes an article in Foreign Affairs. An article is titled "Gene Editing for Good." So essentially, this is kind of the vision of why these technologies are interesting from Gates' perspective. So Bill Gates says, gene editing could help humanity overcome some of the biggest and most persistent challenges in global health and development, thanks to its inherent potential to save millions of lives and empower millions of people to lift themselves out of poverty. Then, I mean, the article goes on. Essentially, Gates also sounds a warning, insisting that, in fact, there are no feasible alternative to this biotech revolution, and that therefore, it says, it would be a tragedy to pass up the opportunity. So that's, I think, how like this technology are envisioned as a way, essentially, to govern global ecologies and make them more apt to operate as a way of capital accumulation, but also as a way of solving all types of like global problems, from hunger to climate change. And I think then Erica, looking at the agricultural side of things, might help us go more into that.
Yeah, I guess the idea was first also to check if like that was a lot to take in.
Yeah.
How are people feeling in terms of the understanding the difference between maybe the older generation of GM technologies and the newer ones, kind of why that's important. You don't need to understand how it works. I think we barely do, but like understand kind of what the political or economic significance is, more or less, of this shift from recombinant DNA to CRISPR. Is that?
No, I didn't get what now we can do that we couldn't do before.
Yeah, I mean, I guess with the in broad terms, or kind of to simplify, the recombinant DNA is inserting necessarily genetic material essentially from one organism to another, whereas with CRISPR, it allows a much broader range of modifications that you can do. So whereas kind of before, okay, you could take a gene from one fish and put it into another fish that might make it grow faster. But with CRISPR, they use language that we're quite critical of, but it's hard to talk outside of that language, talking about switching genes on and off to, for example, encourage certain traits and not others. Or you can introduce multiple traits into a plant or animal. So the biggest difference is really scale, that you can just do much more, much faster, and with a lot less. I think that's the kind of main thing. And as well, I think what we kind of, what we need to know sort of before going into the agriculture bit is about the legal framework, which essentially starting from the 70s that Amedeo was mentioning, these early developments, you have in the US that for the first time the possibility to kind of not patent, but kind of patent a living organism. This was the Plant Variety Protection Act that kind of allowed a company to say, well, I've spent, I don't know how many dollars developing this particular seed. So now, although maybe at the time you couldn't patent it, there was a kind of level of protections that a company could have of their investment. And this all changes in 1980 with the Diamond versus Chakrabarty ruling, it's called, where basically an engineer from General Electric applied for a patent for a microbe that was genetically modified to metabolize oil. So that's where the kind of oil spill kind of part of the intro comes from, that the first ever living organism to be declared a product of human manufacture and that therefore could be patented was one of these microbes. But obviously corporations immediately understood what the implications of this was, which that now a seed, which was for all of human history has been something that is saved and exchanged freely, can now be declared the innovation of a particular company, and it can be patented. And not only can it be patented, you can also be, you as someone that might kind of exchange these seeds with another farmer or with a neighbor, you are now committing a kind of piracy, right? Because this is all under the TRIPS agreement, so the treaty on, oh my God, I always forget basically on intellectual property rights under the World Trade Organization in the mid-1990s, which then extends this patent regime under the World Trade Organization to the whole world. So that's kind of when this first wave that Amedeo was mentioning of kind of first wave of genetic engineering kind of starts to become industrially available. It's because of this patent protection, because no company will necessarily invest all of the money that they need to invest in developing a commercially viable product unless they can kind of stake a claim on it and say that this is mine. Then obviously a huge critique that we have of the patent regime, and we won't go too much into that now, is that obviously all of this knowledge was not produced by these companies. It's kind of socially diffused processes that have taken place over a really long time. So this kind of famous quote by Boris Johnson about the vaccine, that kind of we have the vaccine because of greed, and that's why it makes sense to have patents, because it's what gives incentives to innovate. We're kind of pushing back on that and saying, well, no, I mean, there's loads of problems also with how science is funded and done, but broadly speaking, the knowledge in order to do these things has been built by thousands of nameless individuals throughout the last, kind of 150, 200 years, and is kind of being enclosed through this patent regime by what we'll now talk about is how that's kind of done by agrochemical corporations in the agricultural industry, although there's kind of many other industries where it's being applied. Was there anything else that maybe wasn't clear or that you'd like more info on?
On the
Can we just take the
No, I just wanted to give a super short on the difference between recombinant and CRISPR. I mean, a metaphor that is often used is the one of the word processor. No? So it's like we are in a bookshop. Like recombinant DNA is a little bit like, okay, it allows you to like rip a page from a book and just put it in another book. CRISPR, the idea of the word processor is that in theory, okay, it allows you to kind of like go and change one letter in one book. No? It's just like, it's a red pen, you go, you change one letter. That's like the fundamental, if you want to reduce it. Obviously, it's a metaphor, so it only goes that far. But more or less, that's the idea. No? It's like you can't, you can make more different types of modification. With the recombinant DNA, you can only transfer one, but the page is what it is. You know, it's like, yeah.
Mm.
That actually answers my question. So you can't merge the two. They're not trying to. They don't want to use the two things together. The two types of.
Not really, but you can with CRISPR also kind of insert material, but more or less, like you could have a sequence. I mean, what is DNA? It's a sequence of amino acids. So when we talk about genes, that refers to like a particular part of the DNA molecule that is associated with the production of a particular protein. So it might be known that if you change a C to a T, you're likely to have like another, the body of whatever it is, a plant or an animal or a microbe, will kind of produce its own body differently if you kind of change these sequence of letters, or in this case, sequence of amino acids that make up DNA. Does that make sense?
Yeah.
Cool. I wanted to maybe give some context for maybe why agriculture, I think, is such an interesting way of looking at these technologies. Obviously, we could have chosen the pathway of kind of looking at how this could be used for warfare or all of the things that kind of could go wrong, which we do to some extent. We also focus a lot on the pharmaceutical industry in all of its forms, both the use of genetically modified model animals, so kind of mostly gene-edited mice that are used extensively for medical testing. We talk about the use of genetically modified animals to produce enzymes and proteins, and different forms of gene therapy and human germline editing, which essentially would mean kind of employing these techniques on indirectly or directly on human bodies, with, I think, in our analysis, a lot more implications than simply the things that could go wrong, like beyond the risks of intervening at that level in something as complex as the human genome. Because think about the fact that a bacterial DNA is much shorter than human DNA. And in spite of all of this very confident rhetoric, it's actually very poorly known what the relationships between a genetic sequence and the expression of that is in the cells, especially now many of you might have heard of epigenetics, or kind of, which is now new science for how the environment, so the experience of an organism, might change the expressions of the gene. So, but I think agriculture is really interesting because of how it really is at the base of the capitalist system, and how space is organized under capitalism really was inaugurated with the plantation model in the 15th century, and with all of the like racial divisions of labor and enslavement that entailed, but also the ecologies that entailed. These large-scale, essentially factory farms that are genetically uniform, because it allows you to kind of maximize production, minimize the creativity and kind of input of human labor on the farm. And especially because of the really pivotal role of seeds, really, I think is the most, for me, one of the most striking applications of biotechnology, simply because, as many, especially ecofeminists like Vandana Shiva in her book Biopiracy, really emphasizes how, seed is something that is inherently abundant. You plant a vegetable today, and tomorrow you have more seeds, not less. But with this patenting regime, it kind of breaks that cycle and that autonomy of farmers. But then it has a whole range of other effects. So I think in the book itself, we talk about a lot of different examples, but I kind of want to go a little bit away from just kind of listing at you different ways that this has been employed, although we're super happy to kind of chat about that, but rather starting from the questions that we asked and the kind of main effects of these technologies and the legal regime that accompanies them on the kind of agricultural sector, but also in society more broadly, because it is really a history of enclosure through patents, but also concentration and monopoly, which is related to the patents, because of course a corporation would like to accumulate these patents. The more patents you have, the more control you have over the supply chain and the food system. And also a range of kind of social and ecological effects to do with the types of modifications that are being done, and kind of how, if we try to move away from the fundamental kind of reductionism of genetic science, which really looks at an ecosystem and kind of says, well, the main problem here is that this plant doesn't grow fast enough, or that this plant is, and there's these weeds growing around this plant, and we need to be able to spray it with more pesticides. So obviously these have all sorts of treadmill effects. So I'll kind of half read, half talk you through some of the bits in the chapter on agriculture. Obviously, feel free to kind of ask questions or intervene at any time. I haven't really got an idea of how long this is, so I'm going to kind of improvise it, and then hopefully at the end of that we can also have a bit. Leonie from GM3 will give a little summary of also how this legal situation is changing in the UK, where this is now becoming very much a reality in terms of GM foods in our supermarkets, and also to hear a bit about movements for seed sovereignty from around the world. So the main kind of question that we asked ourselves is, what kind of logic shapes the life forms that are being produced in these laboratories? What kind of mutations are desirable for an agribusiness corporation, as opposed to kind of what might a farmer or small-scale farmer look for? And why are these CRISPR crops and genome-edited animals being represented at once as nothing radically new, so in order to kind of deregulate them and say, well, this is just a continuation of selective breeding, nothing to see here, everything is as it was, but then at the same time as something completely new, revolutionary, that is going to fix all sorts of problems of social and economic crisis, from malnutrition to drought to whatever it may be. So we kind of take a bit of a longer approach to it in the book, so I'm not really going to go into that, probably too long. But the main thing that we kind of wanted to establish before going into the debate around CRISPR is that you can really look at the whole history of capitalist agriculture, especially in the last 200 years, as this cycle of crises and fixes on a very escalating scale. So if you think about kind of the mid-19th century here, with a division of urban and rural areas and the concentration of bigger and bigger populations in the cities and the depopulation of the countryside, one of the effects of this is what Marx calls the metabolic rift, essentially, nutrients in a very basic way being taken from the soil in the countryside and being dumped into the rivers in the cities. And so that then leads to the kind of twin crisis of soil depletion of the earth and nutrient pollution of the waters. And so for the last, since the 1860s at least, kind of finding continuous solutions to these crises has been at the core of the development of capitalist agriculture. So either whether that may be expanding into new areas, or the imperial trade in fertilizer, where Britain imported kind of tons of fossilized bird dung from Latin America in a very imperial kind of uneven trade, and went as far as digging up battlefields in Europe to find nitrogen to replenish the soil with. So that's the kind of longer history. And then one period that we also kind of then jumping forward a bit that we think is important to understand in order to be able to kind of analyze what's going on with biotechnology right now is the period immediately following the Second World War, which is often called, or is called, Was called by this US aid director, I believe, head of the US Agency for International Development, called it the Green Revolution. So I'll kind of read parts of this passage here, where we say that with the end of the Second World War, US hegemony provided the international framework for an imperial extension of industrial monocultures throughout the world. This transformation was partly motivated by the pressing need to offer an alternative model of development to the one advocated by growing socialist movements in the global south. So this William Goud, in a speech which he gave in 1968, said that development in the field of agriculture contains the makings of a new revolution, not a violent red revolution like that of the Soviets, nor a white revolution like that of the Shah of Iran. I call it the Green Revolution. So by that, he's obviously, when he says the developments in the field of agriculture, he's referring to some extent hybrid seeds, but also the use of chemical inputs, so fertilizers, pesticides, etc., and machinery, which won't go into that too much now, but in the immediate post-war period, through US foreign policy, this mode of agriculture was pushed very aggressively across the world with some very wide-ranging material effects in terms of eroding farmer autonomy all over the world and placing more and more power in the hands of the US agrochemical industry. We said that it provided an opportunity to open new markets for the chemical products offered by the US industry, which combined them with cheap financial credit. So there's this debt trap element of it as well. A cheap financial credit and a variety of developmental programs enabled farmers across the world to acquire new capital-intensive means of agricultural production, which were primarily produced by corporations in the global north. So through this green revolution, farms around the world were turned into receptive markets for synthetic fertilizers, herbicides, and insecticides, but also for these hybrid seeds. So obviously the material effects of this green revolution have been very debated. It's not particularly clear-cut exactly, and it's not that easy to measure exactly what the effects might have been. But one thing that is very clear is that combined with then the later development in kind of the patenting regime through the World Trade Organization, which again we have to understand as a product of US post-war hegemony and all of the international institutions set up during the 20th century with all of their contradiction, is that by essentially these global tendencies towards concentration, mechanization, and chemicalization of agriculture that were opened up in this immediate post-war period were further consolidated in the 90s, especially with the TRIPS agreement in 1994. So because that established a global minimum standard for intellectual property protection and thereby internationalized this US model. And so immediately after that, you see this huge wave in corporate consolidation where these big agrochemical companies basically were snapping up smaller companies, acquiring their patents, and just kind of really establishing their dominance in that entire sector through a wave of mergers and acquisitions. These corporate giants kind of got a truly global market as their playing field, as they specifically try to enhance complementarity between proprietary seeds and proprietary chemicals. So I'm going to talk about that shortly. So by then, fast forward to, I think, 2000, you have 6 companies controlling 75% of the global agricultural input industry. So that's a huge concentration. Obviously, the most famous one probably being Bayer Monsanto, which there was another wave of mergers and acquisitions in the 2010s, where Bayer and Monsanto became one company, Dow and DuPont became one company. So that's the kind of situation by the mid 1990s. So in 1996, you have, and maybe you want to kind of summarize the roundup. Basically, you have 2 big new agricultural commodities that come online and that reshape very drastically how agriculture looks around the world, which is Roundup Ready soy, which is a Monsanto product, and BT crops, which is another Monsanto product. And both of these are kind of chemical interventions, with Roundup Ready soy essentially being, soy was the first one, but then now there are many, many different Roundup Ready crops they are basically engineered to be resistant to the particular chemical that is in Monsanto's Roundup, so glyphosate, meaning that you can, whereas before you could only spray a field with pesticide before sowing it, with these glyphosate resistance, you can keep spraying pesticide all throughout the growing cycle, and you don't have to have people kind of mechanically de-weeding. Yeah, do you want to jump in?
Yeah, no, I think we are coming, we are in a moment of transition, in the sense that exactly, if 1994, so it's like we had like 20 years essentially of kind of like growth of the GM model now in agriculture. Like today, roughly like 13.5% of total agricultural land in the world is dedicated to GM farming, no? So to farm mostly, and of this 13.5%, roughly 88% is herbicide resistant. Mostly soy, maize, canola, cotton. So it's 13.5% of total agricultural area is a lot. It's mostly concentrated in the US, Brazil, Argentina, South America in general, and India. But there are new players. Obviously, Europe to a large extent was never exposed to that because of regulations against the GM agriculture, but this is changing now. So that's an interesting point, interesting moment to kind of discuss this. And as Erika was saying, exactly, like the so-called gene revolution of the 1990s, so the introduction of GM crops, and specifically to a very large extent, when we say GM crops, we're talking about herbicide resistant and pesticide producing or pest resistant crops. It was really very much like the continuation and the integration, the completion of that Green Revolution model. Essentially, the Green Revolution was essentially about the transition from small-scale farming to a large extent for local consumption to big farming for export, no? And the concentration and industrialization of agriculture. Now, concentration and industrialization of agriculture, so you have to automate the production process, and very much kind of herbicide resistant and pesticide producing crops like BT crops served that purpose. On the one hand, herbicide resistant crops essentially made herbicides the central kind of chemical in agriculture. I mean, before the introduction of herbicide resistant, of Roundup ready crops, Roundup was a marginal commodity. It didn't have like a huge market. I mean, you could use it before planting the plants, but you couldn't use it during the. So like the centrality of herbicides in production was established through that. And BT crops are very interesting as well, because with BT crops, I mean, you have a pesticide like BT that essentially is the cryotoxin of Bacillus thuringiensis, of a bacteria that was already used before as a pesticide. No, you could spray it on the plants. You would grow these bacteria in a factory, and then you would collect these cryotoxins produced by the bacteria. You would transport them in the fields, and worker would spray them on plants and so on. Essentially, what BT crops, what the recombinant DNA did there is like it brought all of this production process from the factory into kind of like the cell of the plant. No? So the production wasn't done in a factory far away and then sprayed on top of the plant, but the plant itself was transformed in the factory of cryotoxin. Now, the plant, through its metabolism, having been spliced with bacteria, with with the genes from Bacillus thuringiensis was producing as part of its life these toxins. So on the one hand, obviously for agribusiness was like eliminating a lot of labor, essentially. That was like part of the effect. And the other end was like big savings on kind of like logistics and bringing in. Was that? Should I go into CRISPR crops? No.
Maybe I wanted to say something before about before the CRISPR crops that already when all of this was going on in the 1990s, there was a much more extensive and politicized debate around these technologies. During the WTO negotiations, there were broad global mobilizations against this patent regime. It was like very clear that this was a case of kind of corporate enclosure, and that it was something that in many cases in particular would really violate the rights and the kind of life ways of indigenous people all across the world. So for example, in 2001 there was this very widely publicized study that reported that maize fields in Oaxaca in south of Mexico contained genes that were thought to originate from Bt corn, so this corn that produces pesticides in its cells, in the US. And this caused a lot of controversy in Mexico, not only because growing GM crops is not allowed in Mexico, but also because maize is an extremely culturally important food and plant. There is and has been for a long time tens of thousands of different varieties of maize grown in Mexico, whereas in the US you have these gigantic industrial monocultural kind of wastelands. And so what this showed was that you cannot contain these genes, right? They are not isolated, and all of living systems are connected, which is obviously something that indigenous peoples have been saying for a very, very long time. But genetic technologies kind of completely, in their reductionism, in the kind of fundamental and inherent reductionism, are unable to see this kind of connection. So what happened was that then by March 2004, hundreds of farmers entered a meeting organized by the Commission for Environmental Cooperation, and they protested the global trade in GM maize. There's, of course, other kind of political factors around the kind of role of US maize in Mexico to do with NAFTA, etc. But we don't have to talk about that now. So basically, Zapatistas and other indigenous communities based in Chiapas, which is a neighboring state to Oaxaca, set up a seed bank, which they call the Mother Seeds in Resistance, to reclaim indigenous sovereignty over their traditional food. So although the kind of question of where this genetically modified material came from and kind of how it got there was never conclusively answered, this very ambiguity itself kind of continues to be a source of kind of controversy and contention, because this, of course, is happening all over the world, especially in the kind of hotspots for the growing of GM crops. So in the US, in Brazil, in India, and possibly soon here in the UK. So I don't know if Randa, I wanted to ask if do you want to say something about like broader indigenous or like seed sovereignty movements? Or sorry to put you on the spot. We can also wait until the end. Yeah, that's fine. That's fine.
Can I ask you?
Yeah, of course.
Yeah.
Isn't there new developments in CRISPR technology that's quite recent?
Yeah, CRISPR itself is, let's say since 2013, but now there's a new, then there's all sorts of battles ongoing regarding kind of the patents of CRISPR as a foundational technology, and there's also an ongoing kind of regulatory discussion process, which here in the UK, and I think Leonie will say something about that later, about how what this kind of looks like when CRISPR crops might, or basically what the legal framework looks like in the UK today, because this has now kind of been approved here. And the reason why I think we wanted to, there's a political reason for why we wanted to establish a continuity between the first generation and the second generation of biotechnology. Because within the EU and in the UK, they are basically saying, well, everyone was against GMO, there was huge mobilization against it, everyone said it was frankenfood and they wanted nothing to do with it. This is completely different. It's just like these mutations could occur in nature, so therefore they are basically natural, and we don't need to kind of regulate or label these in any way. Whereas we are saying, well, what matters is that these are a product of laboratory, mutagenic, as much of a mouthful as it is, but mutagenic techniques, and therefore it is actually pretty much, you know, even though the technique is similar, it is in a line of continuation with the previous technologies that were regulated much more strictly. So I don't know if you have something you want to...
It's just a case that I used to work in different think tanks, what have you, Whitehall, and you'd hear different people, you know, taking different positions. You'd have different groups, like some people were really icky at the idea of using, you know, genes from one species into another, and yet you had other groups like, I think the one that was most notorious was the group around Marxism today. No, not Marxism today. There's another living Marxism, and they had their own group that was specialised, that did nothing but proselytise the genetic technologies. And you know, so it was like listening to, you know, and it was almost messianic when I was listening to these people. So when you try to get to the core about what matters, you know, like no particular group could give you their own thing, but they couldn't actually give you a meta view, which is quite frustrating.
Yeah, that's very interesting, actually. Yeah, there is definitely even today, but as with many technologies, now there are different positions even in the left-wing movement and so on. I mean, like in the book at some point, I mean, if you take a lot of left accelerationism even today is quite hyped about the potential of genome editing technologies. But I think what was interesting for us is that, I mean, even the critique of genome editing usually emphasizes to a very large extent kind of like the health risks, which obviously is a very interesting and very important debate to have, and we talk about it in the book, and about kind of like the different scales of uncertainty associated with genome editing, not from the uncertainty about the effects it has at the cellular level, but then especially like the uncertainties of how genome editing organisms interact with other organisms more at an ecological global level. No? So that's one element, but then there is an element that is discussed way less. That is kind of the fact that these technologies are developed and are part of much larger industrial systems, and they respond to the logic of these industrial systems, and they reinforce the kind of like, I think on one end they reinforce kind of like global, unequal geographies of production and exchange, colonial geographies of production and exchange, if you want, in the sense that if you, both recombinant DNA and CRISPR technology, to large extent they are dependent on what they're called kind of bioprospecting, sequencing kind of operations. So essentially studying from a genetic point of view organisms from all around the world. You know? That's how like the bacterial enzymes that I was talking about were discovered, no? and so on. So bioprospecting is done all over the world. It's done especially in the global south. But the results of that research are mostly enclosed by biotech corporations in the global north. Then I mean, and then you have inequality also at the level of, so that's the problem of creation of the technology, and then obviously the fact that like a lot of these seeds are patented also creates streams of rent again that goes from the south to essentially to mostly to the north.
I mean, I'm just thinking about time moving on. Does anybody have any questions for the regarding what's been going on so far?
I wanna maybe I'm mindful of time, so we're gonna just kind of give a brief, maybe say something about how we see the main discourse or framing that has been going around CRISPR in agriculture, rather than kind of going into all of the different crops that exist, because there are more than we can count. We have a lot of kind of fun with it in a way in the book, and talking about all of these sometimes quite farcical examples. So I think one of the big kind of aims of CRISPR is how it's being framed as such a promising tool by agribusiness corporations, entrepreneurial scientists, and policymakers, is to kind of secure large-scale industrial monocropping in the face of increasingly insecure and shifting ecological conditions. So advocates of these tools kind of claim that they will finally resolve this kind of contradiction at the core of the food system, by which we have this very jarring juxtaposition of overabundance, scarcity, waste, and famine or hunger. So I think there's this quote from Corteva Agriscience, which is the agricultural branch of Dow DuPont. So big, one of the smaller corporations that exist under the umbrella of a much bigger, giant, mega conglomerate. They frame it in this way. Agriculture is at a crossroads. Plants are under attack from changing weather, drought, floods, heat waves, diseases, and pests. At the same time, our population is growing and consumers are increasingly demanding food that is healthier for their families and the planet. This means that we need tools like CRISPR to grow more food that is better for people and the environment using fewer resources. So this is just one quote of many that basically say this thing, that is like climate change is here, we're facing increasing levels of crop failure, the population is growing, old kind of Malthusian narrative or kind of old eco-fascist narrative around sort of says that people are hungry because there's just not enough food and too many people, which we kind of want to push back against. So basically they're saying there is no alternative. There's something fundamentally kind of neoliberal about this. They say, well, there's no other way that this can be addressed, and rather than kind of the large-scale engineering of CRISPR, of gene editing as a way of kind of addressing the fact that monocultural plantations are not surviving, and a kind of recurrent epidemics, recurrent drought, all of these things that are the accumulated effects of this kind of intensive and monocultural way of doing agriculture. So obviously there's a few different examples that are all really interesting, but I think one, although it's not exactly the most kind of commercially relevant, we still think it's quite interesting and important from its ideological aspects. So one other kind of promise by the biotech industry, including by Bill Gates in the article that Amedeo mentioned earlier, is that it's kind of hunger and malnutrition. So they basically, and by they I mean the International Rice Institute, which is in part funded by the Rockefeller Foundation, quite interesting, and maybe we can have a chat later about like the role of philanthrocapitalism in general in kind of framing the climate crisis and responses to it. But they're basically asking themselves if they can, design charitable crops that can bring nutrients to people that have basically been cut off from the land and that no longer can kind of grow their own food. So, so already in the 1990s, amid this mounting criticism that GMOs were narrowly serving corporate interests at the expense of farmers and consumers, the Rockefeller Foundation started developing what one of their lead scientists described as a purely altruistic use of genetic engineering technology. And essentially what they were trying to do was create this rice that was enriched with vitamin A. And this new GM crop, according to representatives of the Rockefeller Foundation, had the potential to integrate the diet of the global poor. So after years of research, this golden rice, as they call it, it's golden because it's enriched with beta-carotene, which is essentially the things that give the color to carrots. So it has this kind of like orangey tint, was finally flaunted as a ready-made solution to vitamin A deficiency. And so today they have, so this golden rice triggered a series of global controversy. So in 2013, in early field trials in the Philippines, hundreds of local farmers and environmentalists went to do direct action and basically just tear up the plants. And this was supported by one of the largest farmers organizations in the Philippines. And they basically accused the Rockefeller Foundation and other promoters of golden rice of using the victims of poverty and malnourished children as an excuse to promote a product that will pave the way towards more profit and control of biotech corporations to food and agriculture. So, and there's many more kind of, so the idea behind this golden rice is to essentially distribute it. In their case, they're planning to mostly do it in South Asia and Southeast Asia. But at the same time, kind of critiques from all over the world are mounting against this type of approach and basically critique golden rice as extending and deepening kind of a long-standing colonial dynamic. And so over the last 30 years, non-governmental organizations and coalitions and farmers group such as Via Campesina, Movimento dos Trabajadores Rurais Sem Terra, Navdanya International, have kind of challenged these premises of GMO and the way that they entrench corporate power in the food system. So I think one other thing that we kind of want to draw your attention towards is that we mentioned the Rockefeller Foundation quite a few times. They're still very active, and I think it's interesting to kind of be attentive to the parallels between Rockefeller and the Gates Foundation. So while the Rockefeller Foundation was established in 1913, when John D. Rockefeller's like oil wealth was basically the equivalent of something like 3% of US GDP at the time, this foundation was established to kind of to manage and to find ways of diverting like social unrest or like the kind of many forms of dissent that were happening against this concentration of wealth. And in a very similar way, the Bill Gates Foundation has kind of taken up that mantle. And in fact, they collaborate quite extensively. And so, of course, the fact that these are philanthropic organizations doesn't mean that they don't indirectly lead to the accumulation of capital. Because, of course, while Bill Gates is very busy writing opinion pieces about how necessary these technologies are, he's also investing very heavily into them. So the kind of social legitimacy of not obviously the two people in this room, but in broader society that the Gates Foundation might have or that the Rockefeller Foundation might have is then being used to kind of peddle this narrative that these technologies are necessary and therefore also stimulating kind of further investments into them. So I think we can kind of skip over the kind of farm animal bit because we simply don't really have time. But I was wondering if Leonie, maybe would you want to kind of fill us in a bit about the report that GM Freeze has published and kind of what's the last situation in... Oh, so you've spoken right into this the whole time.
No, because I think...
You can use this.
I think we got lost a bit in the history, but it's important to remember that, I mean, in 2023 in the UK it was approved under the Genetic Technology Act that for the first, and in the EU a pretty similar act is going to be approved probably quite soon. With opposition, we'll see. But that opened the possibility of like CRISPR crops and CRISPR animals in the UK. So.
Yeah, so here in the UK we passed the Genetic Technology Precision Breeding Act in 2023, and then the regulations which will operationalise the Act were passed earlier this year. They're going to come into force in November. I'm not sure if this is standard to doing something, yes. Yeah, so hi, I'm Leonie. I work for a campaigning organisation called GM Freeze, and we campaign against genetic technologies. And yeah, it's quite refreshing to come to an event like this, because I don't really get to think about these like more kind of philosophical and like that kinds of questions around capitalism and enclosure of the commons, because my day job tends to be the kind of boring campaigning against deregulation stuff, which is not it's also interesting.
Would you like me to touch it on?
I don't know, I can't quite
I can
hear
keep it
where
there
it's.
for you if
Can
you
people
want.
hear me without it?
Yeah.
Maybe I'll just leave it. So the Genetic Technology Precision Breeding Act defines precision-bred organisms as those that they have been produced through genetic engineering, but they could have happened anyway by traditional processes. So this is the government going, yeah, we're being really pro-scientific. We're going to go with the science and like deregulate all of this technology. But can you think of a less scientific definition that it could have happened in some other way? It didn't happen in some other way. It's called precision breeding, and it doesn't involve breeding, and the definition is completely imprecise. And like that kind of sets off this cascade of like completely anti-scientific like logic, and so I mean the first thing to say about that definition is that it puts developers at a massive legal advantage for like any challenge that like the crops that they're producing couldn't have been produced through another means. Like how do you prove that something couldn't have happened by some other means? It basically means that if it puts developers at a really big advantage. And I went to a meeting of a government advisory body called the Advisory Committee on Releases to the Environment. They're going to be fulfilling a really key role now that this sort of legal aspect has passed. Like they will be deciding what is categorised as a precision-bred organism and what isn't. And so essentially, what's categorised as precision-bred will be in our farms and in our food systems, completely unlabelled, untraceable. We won't know where they are. We won't know that it's on our plates. This is the effect of this deregulation that's happening. And ACRE will decide what is kind of given that categorisation. And so I went to this meeting in July, and they're completely like in a mess, like because of this ridiculously unscientific definition. They've got no means of excluding something from being categorised as a precision-bred organism. And there was kind of one guy in the room who was clearly really uncomfortable. And you mentioned transgenes earlier, like genes from another species. And they were kind of debating this thing called a hairpin genetic change. And they're going, yeah, but hold on a minute, because so he's going, yeah, but this is, if I don't know what a transgene is if this isn't a transgene. And they're like, but that's not the definition that's in the Act. The definition that's in the Act is, could it have been produced by some other means? So we're not even using this like it's genes from another species, you know. And at one point, one of them goes, well, hold on a minute, this definitely couldn't have been produced by another means because it's an entirely synthetic gene. It's like, okay, so this is the power of the technology we're talking about, like completely manufactured and, you know, like human manufactured genetic code essentially, and like can we get that in there and define it as a precision bred organism, and therefore, you know, it's completely unregulated. So I mean, I guess there's kind of loads of interesting stuff that you picked up, that you said that I could pick up on, but also I just wanted to, like, you know, we've did an article here about the about this golden mice thing in the Philippines. It's such a disgustingly colonial mentality around. You know, the Guardian said, "Oh, it's a catastrophe that British, you know, that environmentalists are stopping people in the global south from eating," and it's like. And they described it as Greenpeace. It's like, well, actually, Greenpeace dropped campaigning on genetic technologies years ago because they got accused of being well-fed environmentalists in the global north. And actually it's Greenpeace, the Philippines, it's a really massive social movement in the Philippines objecting to this technology. And yet the Guardian kind of paints it as like, oh, it's just like, you know, northern environmentalists. It's complete rubbish. And it's like, why should people in the Philippines expect to get all of their nutrition from rice? You know, they're perfectly capable of growing carrots as well. Like, and the idea that we should just be like manufacturing crops so that people can have really poor diets is just really it's just on so many levels it's really wrong. But yeah, I mean, there's quite a lot of other interesting technologies around. You touched on a couple, like gene drives, kind of completely wiping out species that aren't very, you know, that cause us problems. You know, mosquitoes being the like was what is quite often raised, but actually, as well as giving humans malaria, they're also right at the bottom of food chains. You know, the idea that we can just like wipe out mosquitoes and then isn't that great for us because we won't get malaria anymore? Yeah, but what about everybody else in the food chain? You know, and is that really a good thing? I think that this kind of lack of systems thinking about the whole, and the lack of sort of awareness of the web of life and how we fit and different organisms interact with each other is just massively arrogant. And yeah, I could talk for ages, but
Absolutely.
I'm sort of
fantastic,
waving some papers around at you. So we've produced this report about the deregulation of newer forms of GM that's on our website, and there's also an article in this newsletter about some of the newer forms of GM, and on it we touch on digital sequence information, which is essentially like stealing the genetic codes of everybody in the world and then being able to kind of use it in some kind of weird AI GM computers designing life forms future, which is not a kind of future that I want to see. But yeah, there's also
we're running out of time. But what I was wondering is if you could get some more copies and then leave them in the shop.
Yeah, sure.
Because we've got some special shelves over there for activists to leave stuff on, and then people can actually use it. But it sounds like we've really escaped the surface of a really complex topic. And I was wondering if maybe this was something that you'd want to continue possibly and say somewhere like the Dalston Solidarity Cafe, where we could have more time to actually discuss it further.
Yeah, it'd be great to continue discussing obviously all of these themes because it's true that it's what we'd hope to kind of have more discussion. There's also simply so much that kind of needs to be said before we can kind of go into discussing what's next, because it's really not that clear cut. I guess I want to offer a couple maybe concluding sort of
I can also...
Yeah. Yes, please. I didn't want to put you on the spot again.
Yeah, no, I know. Yeah, I guess like I don't know. Should I come to here?
Yeah.
Cool.
Yeah, I guess like what so like what you're given is like, and also for me, I feel like I haven't had the chance in the day-to-day job of like working with the Seed Sovereignty program to think about all the broader issues. But I think it's also important, as we've spoken about all the like larger dynamic at play, I think it's also important to bring it back to basics and like why is seed sovereignty important and what it is. And like seed sovereignty is the definition of seed sovereignty is the right of communities, farmers, and indigenous people to share, to save, most importantly, share, sell, and control their own seeds. And protecting like this agrobiodiversity and cultural practices, and like why is it important? Like food system does shape society, and seed is the basis of all life. Like like what all our clothes, all our food starts from seed, like from plants. And with the like at the same time as all these like larger corporations, agrochemical corporations, like taking a hold of all this seeds and like industrial agriculture. At the same time, that has also meant a loss of agroecological knowledge. So like farmers are less and more divorced from these like millennia old practices of saving seed and like just growing in their local conditions. Which means that then they're even more reliant and prey to all of the like because most of the time they are sadly prey to brainwashing by all the lobbyists of agrochemical corporations. And like why is this why is this agrobiodiversity agricultural diversity important? Because seeds, like farmers have been saving seeds for millennia, and this means that seeds are locally adapted to those local and social conditions, because I think it's really beautiful to also recognize how as humans we also are we come into like a more metabolic relationship with these plants, and so that means that they're more like these seeds are more adapted and resilient and resistant to possible extreme climate events as we're coming more and more to face them. Also pest resistance, nutrition. If we're talking about all of these corporations creating, wanting to create more nutritious seeds, we actually have the seeds grown in these agroecological ways are all very nutritious, and also sometimes quite a lot medical, and also it means that they're tastier quite often. I think the thing about seed sovereignty, I think that the thing that brings it home for me is the fact that it really, once you taste diverse seeds and diverse seeds, you realize that it's tasty, and that food, that desire and yearning for food system change can really come from the gut.
I'm afraid we're going to have to finish the talk now. It's 9:15. We've got to shut up the shop. Sorry about this.
Got you.
So let's try and do something about this at Seedosed in Solidarity Cafe, and then bring it more into it.
I'll say a kind of one concluding point, which is the kind of crux that we want to kind of leave people with in terms of kind of the overarching implications of these technologies, which is there is what we think of as a very troubling tension or contradiction at the heart of this science, which is that on the one hand, there is this increasing recognition, even within colonial Western science, that everything is interconnected and that you cannot simply intervene or engineer one variable. So kind of a contradiction between this increasing awareness and ever larger industrial attempts at intervening in that complexity and trying to kind of homogenize and turn kind of the fields and the kind of land into an increasing and more expansive kind of monoculture plantation. But it also goes for the other fields in which these technologies are applied, not just agriculture, but the mosquitoes that Leonie mentioned, or medicine. It all kind of goes very far away from anything that we might call a holistic approach, and kind of tries to intervene in a very technical way on one barrier. So really sorry to everyone about the time that we ran out, especially to you.
It's detail. It's just one of those topics that needs more information. It really does.
It's true.
So can we give it up to...
Thank you.